Okay, so here we are, the second part of protein synthesis. We're going to talk about translation now. So in the first part, we looked at how the messenger RNA molecule, this orange line here with little fingers sticking out of it, was produced inside the nucleus by reading the complement strand from the DNA, which had been opened up at a specific place to expose a gene which codes for a protein. So we looked at that and we saw how the messenger RNA came together. We saw that the little rungs sticking out for the messenger RNA were mostly the same, but very slightly different to the dRNA, in such that the RNA has uracil and not thymine as one of its bases, but adenine, guanine, and cytosine are the same. So we then saw that to get outside the nucleus, the messenger RNA had to be capped and tailed to give it tickets to cross the membrane, and it also had to have the junk DNA, the introns, spliced out. So now we have the finished messenger RNA, which is entered, in this case, just into the cytoplasm of the cell, because this protein is going to work in the cell. And it's been picked up by this purple ribosome. Now, the ribosome comes in two subunits, the large subunits on the bottom here, and the small subunits sitting at the top. In that you can see two tRNA molecules, so it's a little bit obscured by the presence of them. But I've always thought of the ribosome as like just burger buns, with the bottom one being slightly bigger than the top one. Anyway, so ribosome finds this strand of messenger RNA and it picks it up and it just kind of walks along it, reading the base sequences and bringing in tRNA to match them, complement again, and then each tRNA, which is floating freely in the cytoplasm, carries with it its associated amino acid. So depending on the code here will depend on which amino acid that it carries. So research has shown us that the language of amino acids is different, obviously, to the language of DNA, but each amino acid is a letter, and there's approximately 20 of them required in the human body. Now, we already know that the language of DNA and RNA is only 4 letters long. So we've got a little bit of a disparity in the translation. Now remember, this is what we're doing, we're translating. So I've put up here, my translation. So I've got Kia mā ahau, and Kia mā ahau is Scottish Gaelic for how are you? Kia mā ahau, halgama tapalaid. If you're feeling okay, that is. So in the Gaelic language, there are 18 letters. In English language, there are 26. So again, matching things up and making certain sounds in Gaelic requires you to double up letters to create the sounds that we have in English with one letter. The same goes on between the language of DNA and RNA and the language of amino acids. And what we find is that every 3 groupings of base pairs or bases sticking out equals 1 amino acid. There's a certain amount of redundancy in this, because by doing it that way you could have 64 different amino acids and we only need about 22. So without getting into mutations and cancer and stuff like that, which is in part of the PowerPoint lecture but not part of this because it's not key to what we're learning. So we have each 3, and these 3s we refer to as codons, because they code for 1 amino acid. So each of these codons are matched by 3 on the tRNA, which are anticodons, because they're complementary. So we match up tRNA, which is transfer RNA, because it's transferring an amino acid to a ribosome, when the appropriate messenger RNA is sitting in the ribosome. Messenger RNA is there, the ribosome starts here and is moving along this direction. I've put little green lines in here to show where the codons are. So here was the first, here's the second, here's the third, here's the fourth, and here's the two that are working on right now. And you can see 1, 2, 3, 4, which matches these codons. So this part of the protein has already started. Each one of these are amino acids, different amino acids, and they start interacting with each other, and it starts taking on its primary protein structure, which we already looked at back in week 2. So how does this form? Well, you've got your codon sequence here being held by the large subunit. The tRNA is able to come in and slot and match up, and so when the codon and anticodon matches, it's locked in place and held there. The next one is brought in, and the same process goes on until it's locked as well. And because they're locked together, then these two are close together and a peptide bond is formed between them. Now we already looked at that in macromolecules for proteins. So the peptide bond is formed by dehydration synthesis, and then that will be released. This one will move over to this position, and the new tRNA will come in, and then they will be bonded as it moves along until it hits a termination sequence, and then this all breaks off. Messenger RNA is finished, tRNAs break away, and then the polypeptide, which is all of these amino acids joined by peptide bonds, breaks away and is now a primary protein. The secondary structural levels will start to fold with the hydrogen bonds of alpha helices and beta pleated sheets, and then the third tertiary, the tertiary level, the ionic bonds, etc., will start giving it its full shape. There could be some post-translational modifications required as well for it to carry on its job, but right now we're focused merely on how we get this translation from the messenger RNA 4-letter language to the language of amino acids, which is 22. So that's it. The codons are matched up to each three bases on a tRNA as an anticodon that ascribes to an amino acid. They're brought in together to match complementary to the messenger RNA strand. Peptide bonds are formed, polypeptide is formed. So starting at this point we have initiation, just as we had in transcription. As the ribosome moves on, you get elongation of the amino acid, and at the certain point you'll hit the termination sequence, and that breaks off. This is released, and that becomes the protein. And that is how a protein is made from a DNA through transcription and then translation. And as far as we are concerned as physiologists, that is enough. So, we're done.